Welcome. Today we will talk about one of the most interesting chapters in science, and that is cell, the very fundamental unit of life. Now today when we would be discussing about cell, we would be talking about the three major components. Those are the plasma membrane, the nucleus and the cytoplasm. Within the cytoplasm you have various cell organelles that are present, and we'll be discussing those one by one. But before we begin with, as always, we have some of the brain teasers for you, and if you are able to solve those brain teasers, you are all set with the lectures. Else we will come back onto those brain teasers towards the end of these lectures. So in this lecture we would be focusing on what is osmosis, a hypotonic solution, the various components like lysosomes, Golgi bodies, the Golgi apparatus, talking about the various examples for a hypertonic and a hypotonic solution, and so on and so forth. So let's first talk about the very discovery of cell. As we have already covered in class 7th, Robert Hooke in 1665 gave the concept of cell. But during that time, he studied the cork cell, and this was a kind of dead cell from the bark of the tree. Later on, it was for the first time that Leeuwenhoek discovered the first living cell. Very important. So the first living cell was discovered by Leeuwenhoek, and this was seen in the pond water for the first time. Later on, it was Robert Brown who talked about the discovery of the nucleus of the cell. So within the cell you have the cytoplasm and the nucleus, and this discovery of the nucleus was given by Robert Brown. Purkinje gave the term protoplasm, which is the fluid substance that fills the cell. Then you have Sheldon and Shavin, who laid forward the cell theory, which we would study in classes 11th and 12th in more detail. And then you have Virchow, who talked about that all the cells arise from pre-existing cells only, and that was a remarkable discovery of its time, because this was possible only to explain, or this concept led to the explanation that there is evolution, and this evolution occurs only if cell exists from a pre-existing cell. So there was a kind of continuous series of inventions and discoveries that laid forward, and from a simple microscope to a compound microscope to an electron microscope, where you could study each of the organelles, each of the components in a very minute fashion. So as we have already studied in class 7th, we have the unicellular organisms and the multicellular organisms. Unicellular are single-celled, for example, amoeba, paramecium. So these single-celled organisms have just one cell which perform all and every kind of function. However, multicellular organisms like we human have a kind of compendium of cells, and each of the cell is specialized to perform certain action. For example, the nerve cell would do the function of conduction. You would have muscle cell that would help in the contraction of the muscles. You would have RBC, WBC. So all of those are different cells, and all cells are different in shapes and size, as we have already covered in our initial classes. Now, as we discussed, cell is composed of the outer cell wall that is present only and only in the plant cell. So very important question. Many a times you have questions like that. You have onion peel and a RBC. RBC bursts in a hypotonic solution, but onion peel does not. Why is it so? Because RBC is a animal cell, onion peel is a plant cell, and plant cell have cell wall. So a very simple logic to understand this. The next important thing, so cell wall is present only in the plant cell. So the three important components that is present in all the eukaryotic cells are nucleus, the fluid surrounding the nucleus, which is cytoplasm, and then you plasma membrane. Now, as in the human body, we have heart that pumps the blood, we have stomach that eats in the process of digestion. So each of the organs perform different function. Similarly is a cell. Within the cell, each of the organelles that is present perform a unique function, and that special functions helps the life to sustain. So That is a very important prerequisite of understanding this. We would be first talking about the cell membrane or the plasma membrane, then we would focus on nucleus, and finally on to cytoplasm. Within the cytoplasm, we will understand the various cell organelles that are present. So talking about plasma membrane, which is also known as the cell membrane. So cell membrane is selectively permeable. Now there are two functions, diffusion and osmosis. Diffusion, when I say, it's usually the exchange of gases. So what actually happens is the movement of carbon dioxide and oxygen is something that is very, very important. The cell inside has a very higher concentration of carbon dioxide in contrast to the outside, and as a result you have diffusion of gases that takes place, and that leads to exchange. However, when I talk about water, we don't use the term diffusion, we use the term osmosis, and this osmosis is the process of moving from a higher water concentration to the lower water concentration through a selectively permeable membrane, and that's the very basic definition of a osmosis. Now osmosis can be explained under a hypertonic solution, a hypotonic solution, and a isotonic solution. In the initial, when we talked about the structure of the cell, we saw onion peel being stained by, it could be iodine, it could be methylene blue, it could be saffron. All of these stains act as dye and help us to understand the structure of the cell. So we understand the arrangement of the cell in a honeycomb structure. Now in this honeycomb structure, I could say the outermost part is what is the cell membrane. Now this cell membrane, when it is selectively permeable, it helps to exchange gases with the nearby ones. How it happens is basically, I can have a very simple demonstration for you. You have a container. Now in this beaker, you have some raisins that are put in a water solution. After some time, what would you observe? You would observe that these raisins swell. Why does this happen? This happens because this was a hypotonic solution. The concentration of the water outside was much higher as compared to inside, and therefore water moved inside the cell or inside the raisin, I would say in this case, and this is what is an example of a hypotonic solution. On the other hand, you have a isotonic solution. Isotonic solution means the component of water is equal in both the levels, so there is no movement of water. Even if there is movement of the water, the balance is maintained as such. So that is what is isotonic. And the last one is the hypertonic solution. Hypertonic solution implies that you have a solution which is put up in a concentrated sugar or salt solution, and as a result the raisin which is put in a sugar or salt solution which is very concentrated would shrink, and that's because the water would move from the raisin to the outer solution, and this is what is a hypertonic solution. Now I can simply say the absorption of water by the plants is a very good example of talking about osmosis. Similarly, I have an eggshell. Now this eggshell is dissolved in a dilute hydrochloric acid. What would happen is this shell or the egg is made up of, the shell of the egg is made up of calcium carbonate. So the outer skin which encloses the shell would allow the water to pass in, and this would be basically swelling that would be seen. However, when I have a deshelled egg which is put up in a very concentrated salt solution, the water would gradually pass out. So there would be a kind of hypertonic solution, we could say, that would be seen. Similarly, if you are washing your hands with soap, what you would observe in a while is your fingers shrink, or there are folds seen on your fingers. And why is it so? This is because of the hypertonic solution. because of the concentration in your hand is much higher as the concentration of water in the soap, and therefore you have water that moves out and the fingers that start to shrink if those are put in a concentrated soap solution for a longer time. So those are some of the examples where we understand the plasma membrane. Coming on to cell wall, as I already said, cell wall is basically seen only and only in plant cells, not in animal cells, the very first rule. The second rule is most of the cell wall is made up of cellulose and it provides a structural support. Why is this support needed? This support is needed specifically in the cases where we see that plant does not move like animals. So to withstand the extreme temperature conditions, the extreme moisture conditions, precipitation, this has a cell wall to protect. Again, what would happen if the water is being lost from a plant cell through osmosis? The contraction of the cell would occur, but this contraction of the cell would be within the boundaries of the cell wall, and this process is what is known as plasmolysis. So very, very important, the difference between osmosis and plasmolysis. So plasmolysis is a classic condition where the cell shrinks within the cell wall that is there. So I can say shrinking of the cell or shrinking of the cytoplasm rather in a hypertonic solution would be explained as the plasmolysis. Next important thing is a living cell absorbs water by osmosis. So if you have the leaf that is put in a sugar solution, you would see the changes. But if the leaf is put in the boiling solution and then into a sugar solution, there won't be any change because there would be no plasmolysis. Why? Because the cell when put up into, the leaves when put up into the boiling water are already killed. The cells are already killed. Now since there are dead cells, they won't show any kind of osmotic behavior. So osmosis is a classic characteristic of a living cell. Another very, very important concept, and all these concepts that we are talking today could be directly or indirectly part of your conceptual and applicable questions rather. So those are very tricky concepts. If you are very thorough with the concept, you are sure to answer any kind of analytical problems very, very clearly here. Now, as I said, you have something that is swollen. The cytoplasm is swollen. Now this cytoplasm would exert the pressure onto the cell wall. However, this cell wall is not present in the animal cell. So what would happen in the animal cell? In the animal cell, this cytoplasm would burst. But this does not happen in the plant cell because of the support that is being provided by the cell wall. So another very important concept that we have already talked about in the case of onion peel and an RBC example. The next is nucleus. Now within the nucleus you have nucleolus that is present, and this nucleus is responsible for cellular reproduction, where we say the cell has the capability to divide into two. Again, a very important concept, this nucleus is double membraned. So double layered surface of the nucleus is what is known as nuclear membrane. So outside the nucleus you have a nuclear membrane, similar to a cell, where outside a cell you have a cell membrane or a plasma membrane that is seen. Now this nuclear membrane allows the exchange of material within the nucleus to the outside nucleus, and you have chromosomes that are present in the nucleus. These chromosomes carry the genetic information. So chromosomes are made up of chromatids, and chromatids in turn are made up of chromatin. So this is again very, very important. Chromatin forms chromatid and chromatid forms the chromosome. These chromosomes contain DNA, and DNA are responsible for the hereditary information that is carried from one organism to another. In some of the cases, the nucleus is poorly defined, and these are seen in the prokaryotic animals. And in the prokaryotes, what actually happens is, since the nucleus is not well defined, we call it as a nucleoid. So nucleoid is seen in prokaryotes. However, Under eukaryotes, you have a well differentiated nucleus with a nuclear membrane that is seen, and outside the nucleus you have the cytoplasm. Cytoplasm is the fluid that fills the space of the cell, and within this cytoplasm you have various cell organelles that are present. Also, it's very important to note that there are certain organisms which do not have a membrane. For example, viruses do not have a membrane, and therefore they do not show a kind of specific characteristics which are particular to life when they enter into a living body. And that's one of the very important concepts that you would understand as we proceed in the higher classes as well. So very important concepts where we talk about the various organelles, the various animal classification, the plant kingdom and the animal kingdom, and the specifications in each of those. So as of now, the most important thing that you need to understand is the difference between a prokaryote and a eukaryote. Under the prokaryote, you do not have a clear nucleus that is defined, so there is absence of nuclear membrane that is seen, and the nucleus is poorly organized, and therefore we call it as nucleoid, and that's the characteristic of prokaryote. However, in eukaryotes it is well assembled and you have a nuclear membrane that is seen. Coming on to the cell organelles, we have various organelles which are present in the cell. It starts from endoplasmic reticulum. You have Golgi apparatus, lysosomes, mitochondria, and plastids seen in plant cells. So let's discuss these one by one. The first one that we would pick is the endoplasmic reticulum. Now this endoplasmic reticulum is a network of membrane-bound vesicles or tubes or sheets that are present, and these long tubes have a round end. That is something that is a classic characteristic of endoplasmic reticulum. Now this endoplasmic reticulum can be of two types, smooth and rough. The smooth endoplasmic reticulum does not have attached ribosomes, and it's mainly used for the process of storing fat, lipids. So that's the main function. However, the rough endoplasmic reticulum has ribosomes attached to it, and these are the sites where protein manufacturing or protein synthesis, I could say, actually takes place. So the cell membrane is built up with the help of protein and lipids. Protein from the rough endoplasmic reticulum, lipids from the smooth endoplasmic reticulum, and this formation of the cell membrane is what is known as membrane biogenesis. Genesis means to make. So membrane biogenesis is what is seen as a process of making the cell membrane through lipids and proteins. The major function of endoplasmic reticulum, what is it? The first thing is to transport the material between the cytoplasm and the nucleus. The second important thing is to maintain the various biochemical activities of the cell. The third very important function of smooth endoplasmic reticulum, as seen in the liver of most of the vertebrates, is the process of detoxification. So this helps in the process of detoxification along with the manufacturing of fat and lipid. So that's very important part of endoplasmic or the function of endoplasmic reticulum, as you can see. The next is Golgi apparatus. Golgi apparatus was first described by Camillo Golgi, and he basically studied on the nerve cells. While he was studying on the nerve cells, he stained some of the individual nerve cells, and this method was known as black reaction. Now this was called as black reaction, and it was used, this method actually used silver nitrate, and with the help of the silver nitrate you had Golgi apparatus that was discovered. Later on in 1906 he was awarded Nobel Prize for his same discovery and the works on the structure of nervous cells. So that was a remarkable achievement by Camillo Golgi, and the material used was very important. So he used silver nitrate for staining the nerve cells to find out this black reaction through which he late or came onto the Golgi apparatus. Now what is Golgi apparatus? I could say those are membrane bound vesicles which are seen, and they are flattened sac-like structures. They have connections to the endoplasmic reticulum. The main function is packing and dispatching the things to the target. So they pack up the things and dispatch the things to the target inside and outside the cell. Also, they help to make complex sugar from the simple sugars that are present. So formation of complex sugar from a simple sugar is a very important function of Golgi apparatus. Also, this helps to form lysosomes. Now, what are lysosomes? Lysosomes are the suicidal bags of the cell. Now, these are the sacs which are filled with digestive enzymes made from rough endoplasmic reticulum. So, it's a kind of waste disposal system of the cell which is present. Everything that is worn out within the cell goes into the lysosome or the suicidal bag. It converts the complex forms into simple forms, and these are called as lysosomes because they destruct or destroy the complex things and form the simple things. But however, in this process, sometimes there could be a burst of lysosome that could occur. And this digestive enzyme which is present within the lysosome can spread into the cell and digest the own cell and basically finish the cell itself, and therefore they are called as suicidal bags. So if this lysosome in any case bursts and explodes and the digestive enzymes are released outside, it will engulf the cell itself. Okay, so that's one of the most important concepts. So endoplasmic reticulum, we talked about the transport, the support, and the smooth endoplasmic reticulum along with the manufacture of fat and lipid is also important for detoxification in the liver. Invertebrates. Golgi apparatus is important for packing and transporting to the target site. Lysosomes are important as they release the digestive enzymes which convert the complex forms into simple forms. They are also called as the suicidal bags. The next is mitochondria. Mitochondria is called as the powerhouse of the cell. Now what actually happens, it's a double membrane structure. The outer membrane is porous relatively and the inner membrane has numerous folds. These folds are responsible for the generation of ATP. Once the ATP is generated, this ATP is what is known as the energy currency of the cell. And what happens is body uses this energy for making new compounds that are required for the body or any kind of mechanical work that is required for the body. A very important thing to note, besides nucleus, it's only mitochondria which has its own DNA and ribosomes. A very, very important question that you need to know. So mitochondria has its own DNA and ribosomes. Also plastids have, but plastids present only in the plant cell. So again, very, very important question. Now coming on to plastid, as I said, they are present only on the plant, only in the plant cells. They could be either chromoplast or leucoplast. Leucoplast are those which are white or colorless. Chromoplast are colored. Now, if the chromoplasts are green in color, we call it chloroplast, and chloroplastids are responsible for chlorophyll. That's the green leaves where you have the process of photosynthesis that takes place. However, if it is chromoplast, it could be due for the color of the leaves, the color of the flower, the color of the fruit. However, leucoplast is responsible for storage of starch, fat, granules, and a good example of leucoplast is seen in the roots of the plant. So roots of the plant have the purpose of storage, and you have leucoplast which are seen. Now chloroplast, as I said, are responsible for chlorophyll. Now these are the membrane layers which are embedded in the stroma, and they have their own DNA and ribosomes similar to a mitochondria and a nucleus, as we have already talked about. The next important component is vacuole. Now vacuole is a huge empty space, but this empty space is actually the storage sac of the cell. In a plant cell, we say there is one big vacuole which pushes the nucleus towards the side, as we have already seen in class 7. However, in the animal cell you have very smaller amount of and numerous amount of vacuoles that are present. These vacuoles usually occupy 50 to 90% of the cell. The function. First is, it is full of cell sap. So it is responsible for maintaining the turgidity and rigidity, I could say, for the cell. The next is the various sugars, amino acids, proteins, organic compounds are present within the vacuole. In amoeba, food vacuole is there, and this food vacuole is what is through the process of endocytosis you have the food that is taken in. So unicellular organisms specifically have very I could say specialized cells which are present, and the role of these cells is to expel any kind of excess of water which is present and some waste from the cell itself. The next important topic that we would discuss today is cell division. We have covered a separate lecture on mitosis and meiosis. So mitosis is the process of division of cell for the growth of the cell. So you have the mother cell which divides into identical daughter cells for the process of growth and repair. However, when we talk about meiosis, it is half the number of chromosomes being transferred from the mother cell, and this process is mainly responsible for reproduction. So you have offsprings that are generated through the process of meiosis. The plant and the animal cell divide to form the gametes, and after the fertilization you have the offsprings that are present. So under the meiosis you have two consecutive divisions that are seen. The cell divides by meiosis leads to the formation of 4 new cells, in contrast to mitosis where you just have 2 cells which are seen. So that was some of the major things that we have understood. Now coming on to some of the important questions that we were there on the very beginning. So the various cell organelles, you have the membrane structures. So for example, mitochondria has a double membrane, vacuole has a single membrane, and ribosome does not have a membrane. So those are some of kind of important questions. The difference between plasmolysis, osmosis, then lysosomes being called as suicidal bags, because if the lysosome bursts, you have the digestive enzyme that is released and it engulfs the whole of the cell itself. The soap solution example we have already talked about. Again, the next important questions are, bacteria do not have chloroplast, but some of those are photoautotropic in nature. That is performed because of the vesicles which is present along with the plasma membrane in the bacteria. So very important question. And since Golgi body is responsible for packing and transporting to the target sites, the vesicle formation would stop if the Golgi apparatus is removed. We have the examples where we have talked about the hypotonic, hypertonic and the isotonic solutions. Also we have talked about the chromoplast, chloroplast and leucoplast. So those were some of the very important concepts that we have covered in this lecture. We'll be covering many more topics in science. So stay tuned. Have a wonderful day ahead.